So today's our topic of discussion will be the carbohydrate metabolism, that's the glycolysis. Okay, the first part will be the glycolysis. As you all know that in carbohydrate metabolism, there is breakdown of the glucose. Okay, so first of all, the glucose will be break down into the pyruvate, which is known as the glycolysis. Okay, so starting the glycolysis, in definition, degradation or oxidation of glucose or glycogen to pyruvate or lactate. Okay, so in aerobic condition, okay, when there will be the aerobic conditions, okay, in the presence of oxygen, the glucose or glycogen will be break down into the pyruvate. Okay, so when the glucose or glycogen will be break down into the pyruvate, this will be the aerobic glycolysis, and in the anaerobic glycolysis, the product will be the lactate. Okay, so what are the synonyms, matlab what are the other names? First one is the EMP pathway, Amder Meyerhof and Parnas pathway. Okay, so now talking about the types of the glycolysis, so first one is the aerobic glycolysis. Okay, so presence of mitochondria and oxygen. Okay, so in aerobic glycolysis you will see the presence of mitochondria and oxygen. Okay, so I already talked about the oxygen, presence of oxygen, and next the mitochondria presence will be discussed in next part. So in aerobic glycolysis there is absence of oxygen, as already talked about it. Okay, so now talking about the aerobic glycolysis. What is aerobic glycolysis? Oxidation of glucose in presence of oxygen. Okay, when glucose will be oxidized into the pyruvate in the presence of oxygen, and this will be known as the aerobic glycolysis. So and the product will be the pyruvate. Okay, and this pyruvate will be further converted into the acetyl CoA. Okay, so NADH plus H will be reoxidized in mitochondria via biological oxidation. Okay, so in what is anaerobic glycolysis? So in aerobic glycolysis there is oxidation of glucose in the absence of oxygen. Okay, and the end product will be the lactate. Okay, and NADH plus H will be reoxidized by lactate dehydrogenase enzyme when converted from pyruvate to lactate. I think there is no need to write down. Okay, already there is well understood. So next, significance, talking about the significance of glycolysis. Okay, so we are going to discuss about the mechanism of glycolysis. Before that, we will discuss what is the significance of the glycolysis. So first one is the aerobic glycolysis. Okay, so this pathway, it provides energy for all the tissues. Okay, as here is written. So brain derives 80% of its energy from glucose in fasting state. Okay, so you know very well that whenever there is fasting stage, so glucose is stored in the form of glycogen and liver. Okay, so liver, 80% of energy will be derived from there. Okay, so and next one is the anaerobic glycolysis, and this the source of energy for erythrocytes and lens. Okay, so anaerobic glycolysis, that is the lactate formation. So here the source of erythrocytes and lens, okay, energy. So next is the this is also the source of energy for vigorously contracting muscles. Okay, so you know very well when you do exercise very heavily, so you will felt short of oxygen. In that way the anaerobic glycolysis pathway works. Okay, you mean there is formation of lactate in the muscles. Okay, so next is the it provides intermediates for other pathways, that is the 2,3 bisphosphoglycerate for release of oxygen in RBCs. Okay, we will discuss in next slide. DHAP for DHAP for TAG synthesis. Okay, we are also gonna discuss about it, and last one is the pyruvate for alanine synthesis. Okay, so we are gonna discuss each and every point in the upcoming slides. So next is the clinical significance of the glycolysis. Okay, so here are the points for the clinical significances. Okay, I hope you will read these by your own, because our main goal is to discuss about the mechanism. Okay, so next, moving to the next slide, transport of glucose in the cell. Okay, so you know very well, first of all, I will explain you that what is the basic criteria of carbohydrate metabolism. Okay, so here you will see, okay, carbohydrate, I'm labeling it like CH, carbohydrate rich diet. Okay, carbohydrate rich diet are nutrients. Okay, when you will take inside your body, okay, it will go inside your stomach. This is your stomach. Okay, here what will happen, the disaccharides will be converted into the monosaccharides, okay, with the help of the enzymes like sucrose, lactose and maltose. Okay, they all are disaccharides and they will be converted into the monosaccharides. Sucrose will be converted into fructose and sugar. Okay, this will be converted into glucose, and this will be glucose and galactose. Okay, so these are the monosaccharides. Now the glucose, okay, now talking about the glucose part, especially about the glucose part. Okay, other one is the fructose and galactose. Okay, we will discuss also about this. So first one, the glucose from this will go to the liver part. This is your liver. Okay, this is your liver. So when the glucose goes through this, the hepatic portal vein, you can see, to the liver. Okay, so what will happen in the liver, the glucose will be converted into the glycogen. Okay, glycogen, the extra glucose, okay, that is not needed in the body at that time, that glucose will be converted into glycogen. Okay, so when glucose will be converted into glycogen, so this process is known as glycogenesis. Okay, I hope you know this term, glycogenesis, conversion of glucose to glycogen, and conversion of glycogen to glucose, this is known as the glycogenolysis. Okay, so next part is the, now this glucose, then glucose is converted to glucose. Okay, glycogen will be converted into glucose. Now this glucose, here you will see the glucose, this glucose through the blood. Okay, this is the blood vein. Okay, so I will label this with the different color. This is the blood. Okay, so when this glucose will go through the blood to the, where it will go to the cell, this is cell. Okay, so inside the cell, the glucose metabolism will take place. Okay, so how this glucose will be transported to the cell? Okay, there are presence of some ग्लूकोज ट्रांसपोर्टर्स. ओके, यू कैन सी द ग्लूकोज ट्रांसपोर्टर, ग्लूट जिनको हम कहते हैं. ठीक है, GLUT, ग्लूट ट्रांसपोर्टर्स. ओके, सो देर आर नंबर ऑफ ग्लूट ट्रांसपोर्टर्स इन साइड अवर बॉडी. GLUT1, GLUT2, GLUT3, GLUT4, and all play a different role in different places. Okay, role will be the same, their location will be different. Okay, so GLUT1, GLUT2, GLUT3 and so on. Okay, so now we will discuss about how the transport of glucose into the cell will be take place through this GLUT. Okay, so finally we will discuss this section. So here you can see the transport of glucose into the cell. How this is glucose. Okay, this is extracellular space, this is cytosol inside the membrane, and you can see here the glucose transporter 1, that is GLUT1. Okay, so glucose here it is entering inside the cell like this. Okay, and here you can see the glucose transporter state 2. Okay, so state 2. This is about the state 1 and state 2. You can see the conformation here and also the conformation here. Okay, so there is change in the conformation, and the glucose molecule is transported from outside to the inside and the cell finally. Okay, and the GLUT2 transporter is involved here in the cells. So next slide is about the metabolic site of glycolysis reactions. So here you can see all tissues, main metabolic pathway giving energy, erythrocytes and nervous tissue drives the energy from glycolysis. Okay, this is very important, that the erythrocytes, that is red blood cells, and nervous tissues, okay, so they derive the energy from the glycolysis. And the cellular site will be the cytosol or cytoplasm of the cell. Okay, so now discussing about the three phases of the glycolysis. Okay, so phase 1, phase 2 and phase 3. Phase 1 will be the phosphorylation of hexose sugar, phase 2 will be the hydrolysis of hexose to triose, and third will be the oxidation of the triose and energy capture. Okay, energy spending phase. Okay, here the energy will be utilized, energy, that is ATP, will be utilized in this phase. In second phase, there is a splitting. Okay, there is splitting of the product. And the third one, here there will be the energy releasing first phase. Okay, so talking about the phase 1 glycolysis, here you can see, okay, so glycogen, as already discussed in this in my slide, that glycogen will be converted into the glucose. You can see here. Okay, so glucose from blood is converted into glucose 6-phosphate. This is the step number 1. Okay, so with the help of magnesium ion, okay, and here in energy in the form of ATP, okay, the glucose will be converted into glucose 6-phosphate. Okay, so here you can see 1 ATP is spent. Okay, so we are also seeing the regulation mechanism. So here you can see there are negative regulators and positive regulators. Okay, so what does it mean? Negative regulators means that in the presence of these three, citrate, glucose 6-phosphate, glucagon, there will be inhibition of this ATP, this glucose conversion to glucose 6-phosphate. Okay, so these are the negative regulators, and insulin and glucose are the positive regulators. It means that these glucose will be converted into glucose 6-phosphate successfully. Okay, so this is the step number 1, and glycogen will be converted to glucose 1-phosphate here. Okay, so here the use of the glycogen phosphorylase enzyme is there, and in second step, glucose 1-phosphate will be converted to glucose 6-phosphate. Okay, so in the first step, the ultimate product is glucose 6-phosphate. Okay, so first glycogen will be converted into glucose 1-phosphate. Okay, and finally it will be converted into the glucose 6-phosphate. And when there is glucose from blood, so it will be directly converted into glucose 6-phosphate with the help of magnesium ions and ATP molecule. Understood? And next phase is about the glucose 6-phosphate. Now this glucose you can see here, this is a irreversible reaction. There is no reversing of the reaction. Okay, so this is the, you can see that committed step. Okay, no reverse reaction will take place. What here you can see, this is reversible reaction. So glucose 6-phosphate will be converted to fructose 6-phosphate, and it will reversibly converted to glucose 6-phosphate. The enzyme involved here will be the phosphohexoisomerase. Okay, understood? So you can see this is aldose sugar and this is the ketose sugar. So there is change in sugar, okay, with this help of this enzyme. Okay, so fructose 6-phosphate, there is also a source of forming this sugar, that is when is the excess of the fructose in our body, so the fructose will be converted to fructose 6-phosphate with the help of enzyme hexokinase. Okay, ATP to ADP. So here you can see the glucokinase and hexokinase. Okay, so glucokinase it will be involved, its role will be in the liver, and in other cells the role will be of hexokinase. Understood? So next step is the fructose 6-phosphate. This fructose 6-phosphate will be converted to fructose 1,6-bisphosphate, okay, with the help of same magnesium ion. Okay, as I'm proceeding into the further steps, please note down how many magnesium ions are involved, how many ATP molecules are forming, are splitting, and what are the enzymes that are involved in the process. Okay, in the end you have to summarize all this. Okay, you have to sum up all these cofactors. Okay, so this magnesium ions is also involved here, and phosphofructase kinase 1. Okay, so here this is the irreversible reaction. Okay, so regulatory step you can say. Okay, so ATP is converted to ADP, and fructose 1,6 bisphosphate is formed here, and you can also see the ATP is also spent here. Okay, so 2 ATPs are used till now. Okay, so negative regulators are same glucagon, citrate, ATP. Okay, please remember these are very important, that these are the negative regulators, and positive regulators are ADP, fructose 2,6 bisphosphate, and insulin. Okay, so next step is the phase 2 glycolysis. Here you can see the fructose 1,6 bisphosphate in the step 4 with the help of this aldolase will be splitting into the, this is the splitting phase, right? So splitting into glyceraldehyde 3-phosphate and dihydroxyetane phosphate. Here you have noted that synthesis of TAG a step was also there. Okay, so here the synthesis of triacyl glycerol takes place from this dihydroxyacetone phosphate. This is very important, and this glyceraldehyde 3-phosphate, they are interconvertible with the help of the enzyme triose phosphate isomerase, and glyceraldehyde 3-phosphate will be converted to glyceraldehyde 3-phosphate, 2-glyceraldehyde, you can see here. Okay, now glyceraldehyde 3-phosphate with the help of glyceraldehyde 3-phosphate dehydrogenase 1,3-bisphosphate residue. Okay, note down the enzymes involved here are very important to remember, and the cofactors, the ATP units are also very important. Okay, so you have to remind there how many units are involved there. Okay, be careful about each and every point. Okay, so here 1,3-bisphosphoglycerate is formed from glycerate 3-phosphate. You can see the hydrogen will be, there will be addition of hydrogen in NAD. Okay, and NADH will be formed, that is plus H positive. So oxidation reaction in glycolysis, this undergoes the oxidation. You are also watching here. Okay, because hydrogen, there is loss of electron here also, so oxidation reaction is involved. Okay, so you can see the iodoacetate, arsenate, irreversible inhibitor. They are the arsenate and iodoacetate, these are the irreversible inhibitor here. Okay, and there are limited NAD in mitochondrial oxidation, and you can see that 1 NADH will be equal to 2.5, and 2 will be equal to 5 prime ATP, right? And now this 1,3-bisphosphoglycerate will be converted to 3-phosphoglycerate here with the help of same magnesium ion, and here ADP will be converted to ATP. Okay, so this is substrate level phosphorylation. You can see that here the addition of phosphate taking place. Okay, 2-phosphate ATP are formed, you can see. So now here there is another step, 1,3-bisphosphoglycerate with the help of this mutase enzyme in RBC, they will be converted, they will be in RBC it will be transformed, okay, 2,3 bis phosphoglycerate. So in RBC what happens, there is loss of ATP and it helps in the dissociation of oxygen from hemoglobin. What is this role of the 2,3 bis phosphoglycerate in RBC? You can see here, okay. So this pathway is known as a Rappaport-Liouvering shunt. This is very important to remember. And here 2,3 bis phosphoglycerate with the loss of the phosphatase, with the loss of the phosphate ion, will be converted to 3-phosphoglycerate again. So now 3-phosphoglycerate will be converted to 2-phosphoglycerate, and it will further converted to phosphophenyl pyruvate, the loss of water with the help of enolase and phosphoglycerate mutase, okay. So here the negative regulator will be the fluoride, okay. Fluoride, you know very well, they are present in the toothpaste, mouthwashes. I know that you know this, okay. So next is the phosphophenol pyruvate will be converted to enol pyruvate, and enol pyruvate will be further converted into pyruvate, okay, with the help of this water, and same ADP will be converted to ATP, to ATP. Got it? Okay. So the another substrate level phosphorylation is taking place here in this step, okay. So phosphophenol pyruvate with the help of magnesium and ATP will be formed. So with the help of this pyruvate kinase, okay. So you can see the positive regulators are fructose 1,6 bisphosphate, okay, and allosteric inhibition, if you see here, is of ATP alanine activator, and these are the inhibitors, and activator will be the F1,6 bisphosphate, okay. So hormonal inhibitors are glucagon, and activator will be the insulin, okay. What does this mean? That a glucagon, when there is increased level of glucose, okay, so when there is increased level of glucose, the insulin will be get activated, and when there will decrease level of glucose, glucagon will be activated, okay. I hope you understand what I'm trying to let you understand. So here you can see the pyruvate kinase enzyme deficiency in RBC leads to decreased ATP formation, altered RBC membrane, phagocytosis, and clinical picture एनीमिया, ओके. एनीमिया कैसे होता है ये आपको यहाँ पे बताया हुआ है, ठीक है. सो नाउ टॉकिंग अबाउट द रेगुलेशन ऑफ द ग्लाइकोलाइसिस. सो रेगुलेशन ऑफ ग्लाइकोलाइसिस, देयर आर 3 स्टेप्स, ठीक है. हेक्सोकाइनेस, द वेरी फर्स्ट स्टेप वाज हेक्सोकाइनेस, एंड दिस इज द फर्स्ट स्टेप ऑफ रेगुलेशन ऑफ ग्लाइकोलाइसिस आल्सो, okay. So here the feedback inhibition or glucokinase insulin, understood. Second is the regulation of phosphofructokinase, okay. Second step, regulation will take place at this step, and third one is a pyruvate kinase, same, okay. So here is the overview. I don't think that I have to repeat it, okay. You please repeat or read it by your own. So next is the energetics of the glycolysis. Energetics, what does it mean? That how many molecules, different type of molecules are involved here. So ATP used, you can see how many ATPs are used and ATP synthesized. One side you will see ATP used. When you will see at the prep stage, there will be use of the ATPs, okay. And in the post prep stage, you will see the synthesis of the ATP. So glucose to glucose 6 phosphates, 1 ATP here used, fructose 6 phosphate, 1 ATP is used here, total 2 ATP, and here also you can see the 2.5 ATP in this step, okay. I already taught you there. And 1,3 bis phosphoglycerate, 2,3 phosphoglycerate, here 2 ATP were involved. So 2 ATP they all got cancelled, and finally 2 ATP was the product, okay. They both got cancelled, okay. Here the ATP were used, and here the ATP synthesis, and both got cancelled, but this 2 ATP will be the whole product. Okay, so alternate fate for the pyruvate, this is very important. Pyruvate, as you saw, this was the end product of the glycolysis. Now there are alternate fate of the pyruvate. Okay, so alternate one is the transamination. Okay, when there will be transamination of pyruvate, there will be formation of the alanine, right? And when there will be the fermentation, ethanol will be formed, and these are found in the microorganisms. Okay, and next is the, and there is formation of acetyl CoA from pyruvate with the help of pyruvate dehydrogenase complex. Okay, so here you see the NAD will be converted to NADH, and reversibly same NAD. Okay, and here the evolving of the CO2 will also take place here. Okay, so here you are watching the reversible step, that is the pyruvate is converted into oxaloacetate, and oxaloacetate is reversibly converted into the pyruvate with the help of this pyruvate carboxylase enzyme. And here you can see there is use of the carbon dioxide. Okay, this is very important to remember, that what are the enzymes that are involved, and what are the other factors that are also involved. Okay, so here you see the third one, NADH will be converted to NAD, pyruvate will be converted to lactate will be this, and this is also known as this step is also known as anaerobic glycolysis. Okay, so oxidative decarboxylation of pyruvate to acetyl CoA. Okay, please leave this. Next one is the reduction of pyruvate to lactate. Okay, anaerobic glycolysis. So pyruvate will be converted to lactate reversibly, and here NADH will be converted to NAD, and NAD reversibly converted into the NADH plus H with the help of this lactate dehydrogenase. Okay, so end product of anaerobic glycolysis, talking about this, so major end product will be found in the lens, kidney medulla, testis, RBC due to poor vasculature. Lactate dehydrogenase regenerates NAD. Okay, it regenerates the NAD. So in skeletal muscles, NAD's production exceeds oxidative capacity. Okay, so there will be the exceeded production of this NADH. So hence reduction of pyruvate to lactate will takes place. Okay, and but if accumulate cause lactic acidosis, when there will be much accumulation of lactate, so there will be the muscle cramp. Okay, and to this reduce the level of the lactate, Cori's cycle will be operated. Okay, so shunt pathway I already explained in upper section. Here you can see in this wait a second. Yes, I have this labyrinth shunt. Okay, I already explained this is what happens in RBC. How does 2,3-bis phosphoglycerate work, how it interferes with the binding of oxygen with the HP. So I hope you understood. Yes, I have explained all this. Yes, again this slide, and yes, the significance of 2,3-bis phosphoglycerate, this is very important. Okay, so there's no net ATP formation. Presence of 2,3-bis phosphoglycerate allows dissociation of oxygen at tissue level with PCO2 is high. I already explained, I think. Okay, so where partial pressure of carbon dioxide will be high. Okay, when there will be high binding of carbon dioxide with the blood, so you will see that 2,3 BPG will disassociate the oxygen from the blood. And finally, thank you.